Enteric pathogens cause a local inflammatory response and the development of protective immunity. In spite of the array of innate immune mechanisms in the gut and stiff competition from the indigenous flora, the gut is a frequent site of infection by pathogenic microorganisms. These include many species of viruses, enteric pathogenic bacteria, including Salmonella, Yersinia, Shigella, and Listeria, and protozoa such as Entamoeba histolytica and Cryptosporidium. These organisms cause disease in many different ways, but there are certain common features of infection that are crucial to understanding how these pathogens stimulate an immune response by the host, in contrast to the immunological tolerance shown to the foreign antigens ingested in food. The most important consequence of infection in the gut, as elsewhere in the body, is the development of an inflammatory response. The release of cytokines and chemokines in this response is key to the induction of an adaptive immune response. The inflammatory mediators stimulate the maturation of dendritic cells and other antigen-presenting cells so that they express the co-stimulatory molecules that provide the additional signals for activation and expansion of naive lymphocytes. Some intestinal pathogens infect enterocytes, the absorptive cells that line much of the intestine. Enterocytes do not act as passive victims of infection, but signal infection by releasing cytokines and chemokines. These include the chemokine interleukin-8, which is a potent neutrophil chemoattractant, and CC chemokines, such as MCP1, MIP1 alpha and beta, and Rantis, which are chemoattractants for monocytes, eosinophils, and T lymphocytes. In this way, the onset of infection triggers an influx of inflammatory cells and lymphocytes, leading to the induction of an immune response to the antigens of the infectious agent. Injury and stress to the enterocytes lining the gut may also stimulate the expression of non-classical MHC molecules, such as MICA and MICB. These act as ligands to the receptors on the gamma delta T cells at the base of the crypts, which kill the infected mucosal cell, thereby promoting repair and recovery of the injured mucosa. And that's enteric pathogens cause a local inflammatory response and the development of protective immunity.